my wonderful PU2 champions. I hope my lovely children are doing absolutely great. So our FA2 examination just got over. I hope that for everyone it was nice. If at all it was not that great, it's okay. You still have chance. You have your next examination coming up. You can give your best effort and see what is the best result when you give your best. Okay, so I am here to help you with that. Today we are going to solve the model question paper number 2. I just want one promise from you. Probably I'll give you a challenge. The challenge is that for the next 30 minutes, okay, you should not be focusing on anything apart from this video. This video should be something you should have full focus on so that we can learn new concept, solve the questions together, and my dear children, remember things for a longer time. Do not make any notes, just listen to me. It should be just you and me. Done. And after 30 minutes, you probably can pull out a paper and take down your notes, or probably can, go around the model question paper if at all you have, and mark it in that these questions are being done. I know the answer. Try to solve it and see if at all you know the answer for this now. Okay, so I hope if this video helps you. Well, my dear children, wishing you all the best for your examination. Let's go ahead with the first question. This is the 1 marker question. Statement 1. Apomixis is the production of seeds from unfertilized ovules. Is it? Apomixis is the production of seeds from unfertilized ovules. How is seed being formed? So basically, we have the ovary. Inside that we have a structure known as ovule. Inside the ovule, so I'm writing ovule, okay? Inside the ovule, we have a little structure that is known as embryo sac. And within the embryo sac, we have the female gamete. This is what? The female gamete. Now, when the male gamete comes, when the male gamete comes, male and female gamete will be fusing together to form a zygote. So female gamete, as you know, all the gametes are haploid in nature. They will have only half the number of chromosome. So they have N, half number of chromosome. Another half is given by the male gamete. Together it becomes 2N. And that's how you get a zygote. And when you have a zygote being formed inside the ovule, you will now not call it as an ovule. Rather, you will now call it as what? A seed. It has formed a zygote. The zygote will soon transform into embryo. The embryo will soon transform into what? A whole new plant out there. So it's a seed now, my dear children. Now over here it is said apomixis is a production of seeds from unfertilized ovules. So this process is known as fertilization, where this and the male gamete fuses. You call this as fertilization. Now I am telling you that this process means the seed is being formed from unfertilized ovule. Do you think without fertilization seed can be formed? Arey Baba, how will zygote be formed? So the answer is yes, it can. And this exact process is known as apomixis. So what exactly happens is, usually the female gamete is haploid in nature, right? But over here, due to some mechanisms out there, it is not haploid, rather it is already diploid in nature. It is already diploid in nature. So it does not need to wait for the male gamete to become 2N and become a zygote and all. The female gamete transforms into the zygote. It will be transforming into the zygote. And now when you have got the zygote within the ovule, you will now anyway call it as a seed. You get it? But fertilization is not happening. So apomixis is the production of seeds from unfertilized ovules. This is a correct statement. Statement number 2. Embryos produced from apomictic seeds are not generally identical to the parent plant. Now the embryos which are going to be formed, they are not going to be identical to the parent plant. Do you think so? Arey Baba, whatever gametes, whatever genetic makeup is there in the female gamete, the same thing is transferred to the zygote. Right? So obviously it has to be identical just like the parent, my dear children, which is why we say that it is type of asexual reproduction. It mimics asexual reproduction. In asexual, only one parent is involved, right? And whatever things are being formed, they are all clones. They all look like each other. They are what? Clones. So over here they are saying it's not identical. This is wrong. So statement 1 is correct. Statement 2 would be wrong. Statement 1 is true. Statement 2 is false. Where do you see it? Apomixis is usually seen in Asteraceae as well as grasses. Asteraceae and grasses, always remember. Why is it useful for the farmers out there? Because my dear children, suppose this plant who produces the female gamete has good amount of features, is pest resistant, is drought resistant, this and that. So whatever features are there can be transferred to the zygote, and the zygote would be exactly like the parent plant. So it can survive better. Okay, so your answer for this question is A. I hope you got it. And if you think you are understanding things well, first of all, share it with your friends. Second thing, click on the subscribe button right now, because the video is not limited to this, but so many other contents as well, which is definitely going to help you out. Okay, question number 2. The cells that secrete testicular hormones are testicular hormones. That means the male sex hormones, you also call them as androgens. The most famous androgen that we have is testosterone. But what is the cell that releases this androgens, my dear children? So you know what? Inside the testis, suppose this is a testis. Inside the testis, you have testicular lobules. What do you have? Testicular lobules. Inside that, you have seminiferous tubules. How many? Almost 2 to 3 seminiferous tubules per the testicular lobules. So if at all I zoom it in and see, so what is exactly happening? So this is a seminiferous tubule. This is another seminiferous tubule. Okay, inside the seminiferous tubule, my dear children, you have two things. One is Sertoli cell and other is your spermatogonia. Spermatogonia, that is the germ mother cell. It will divide to form the sperms later on. But who provides nourishment? The Sertoli cells provide the nourishment right over here. Okay, just give me a second. It makes me run sometimes. Now, why is ma'am telling me all this thing? So basically, listen. So this is happening within the seminiferous tubules. Now, in between this, you see a space. You call the space as interstitial space, or you also, you call the space as interstitial space. And over here you have some cells, and you call these cells as Leydig cells. What do you call? Leydig cells, or you also call them as interstitial cells. What do? They are the one that releases testicular hormones, which gives the male their secondary sexual characters. Now this testicular hormone, which is going to be released by the Leydig cells, are super important for the process of what? Spermatogenesis. That is the process of formation of sperms. So over here, the cells that secrete testicular hormones are Leydig cells, not Sertoli, because they are for nourishment. Germ cells basically are gametes. Spermatogonia is the germ mother cell. That means it divides to form the germ cells later on. Your only answer for this question would be B. Cool. Let's go ahead. Which of the following is not a copper releasing IUD, intrauterine device? Which one is not a copper releasing IUD? LNG20. The first option itself is your answer. My dear children, copper releasing IUD, do you know how does it work? So basically, suppose we have a structure that is known as copper T. On this, we have the copper wire being enrolled, and this is put inside the uterus. So what exactly happens is that, suppose a sperm tries to enter, okay, the copper ions would be acting on the tail of the sperm and does not allow the movement of the sperm. If sperm cannot move, do you think it can go and fertilize? No. And also it reduces the fertilizing capacity. That's the exact thing, how it works, and it does not cause much trouble to us, my dear children. It's pretty much safe. And it's an intrauterine device. It's put inside the uterus by the doctors. Okay. So CUT, that is copper T, is an example. CU7, it looks like a 7-shaped structure. That is why you call it CU7. Multiload 375 had that much amount of copper being released, which is what is known as multiload 375. LNG20, my dear children, it is levonorgestrel 20. That is a synthetic form of our hormone progesterone. Our hormone progesterone. So progesterone, which is in our body, it usually helps in the process of ovulation. That means for the release of egg. It usually helps in the process of implantation. It makes the endometrium thick and everything out there. But over here, whenever we are giving synthetic hormones to our body, the level of hormones would be obviously hampered. It's not now normal. So what will happen? Everything that it was doing, it will start doing the opposite of it. Ovulation is not happening. Implantation would not happen because very thin endometrium would be there, and also there will be a mucus plug being formed, such as sperm cannot enter through the cervix. So LNG20 is your answer. It is a hormone releasing IUD. You have multiple type of IUD. One is non-medicated IUD that does not release anything. Okay, the second one is copper releasing IUD. The third one is what? The third one is hormone releasing IUDs. So LNG20 is your answer. Next, what is the possible blood group of an offspring when both parents have AB blood group? Baba, when both the parents have AB blood group, AB and AB, what are the possible offsprings, or what do you call possible blood groups in the offsprings? This and this fuses, you get AA. AA is there, that means A blood group. This and this fuses, you get AB. AB is there, both of them show co-dominance. Both of them are equally dominant, so which is why we get AB blood group. This and this fuses, again AB blood group. Okay, this and this we get BB, which means it will be B blood group. So over here, how many types? Do not count it twice. Okay, count it only once. AB once. Different types you are being asked. You are not asked how many number of offsprings are being produced, right? So one type, second type, and the third type. A, AB, and B. So your answer for this question becomes B. Cool? Yes, ma'am. Now, if an mRNA 3-AUG undergoes translation, how many types of amino acids are present? If this particular mRNA undergoes translation, how many types of amino acids are going to be present? What do you think so? So ma'am, we know, so it comes in the codon, right? So 3 of them, 3, 3, 3. So if at all you see, AUG is for methionine, so this is methionine. UUU is phenylalanine, it's same. It's same. So we do not count it twice, ma'am. And last one, UAA is a stop codon. So basically just 2 types of amino acids are being produced. The answer is wrong. Why? Translation, okay, translation happens in 5' to 3' direction, not from 3 to 5. So basically you have to think in this aspect. Do not go around like that, my dear children, and that's how we lose our marks. So basically if we see, we start from here. AAU, one type of amino acid. UUU, second type of amino acid. This is the same. This is the same. We do not count it. GUA, this is third type of amino acid. None of the stop codons are being mentioned over here. So how many types of amino acid? 1, 2, 3, ma'am. 1, 2, 3. This is the mistake that children do. It's a very easy question, but we tend to do the mistake, and it's okay. Even I do the mistake, okay? So it's not like you are alone in this. We all do mistakes, and we should be learning from mistake. Okay, so this is what you need to remember every time you have a transcription related question. Check the polarity. Cool, let's go. Now, first of all, if at all you are someone who is preparing for KCET examination, let me tell you, we have already launched our ABHYAS KCET test series, one of the most famous test series out there, for good reasons, obviously, because so many children utilized it and got the benefits of it. All of them, most of them have got what? Not all of them. Most of them have got what? Under top 1000 rank. And here there were so many children who came to meet us as well. We have Prajwal with rank 171. Out of 3 lakh children, just imagine if someone is scoring 171. Obviously their hard work and obviously the resources they are referring to, and this is one such resource. We have Kiran with 443 rank, we have Deeksha 693, Sachin with 934, Sahil with 2427. So many children, my dear children. So we thought, why not launch it early? We did it a little late the previous year. We have launched it early now, but this is the December one, and we do not know when it is going to come again. So if at all you have not registered, make sure you do it quick. You'll get 35 KCET test, okay? And my dear children, please understand one very important thing. You will be having chapter-wise KCET PYQs in the test format. So this is an addition. We have not changed the price. The price is kept to the same, but we have added up one more thing, thinking it will be useful for you. So there will be KCET chapter-wise questions, as in it will be given in the form of a quiz. You can go and attempt it, okay? There will be real exam pattern and timing, as I said, for all the tests out there. It will have full PCMB coverage, including 12th grade, or any, sorry, not 12th grade, including PU1 and PU2. The price for this particular thing is 1499. Go and take it. That's the only thing that I want to tell, if at all you're preparing for KCET examination. And let's go ahead with the next question. Arrange the following animal groups in correct sequence of their occurrence on geological time scale. But before that, my dear children, please do it quick, okay? If at all you're taking ABHYAS, take it quick and start solving questions. Then only there will be use of it. If at all you are buying it and not using it, it's of no use, right? So please start using it. It is going to definitely help you out. Yeah. Now, there are animal groups given over here. You have to arrange them in the order of their geological time scale, as in when who was the first organism to come from them, who came, and everything like that. So my dear children, the first organism that came among them is fishes. From fishes, okay, fishes were in water. Then we got organisms that could live both in water as well as land, that is amphibians. From amphibians, we slowly got reptiles. From reptiles, we have 2 divisions. One is aves, that is birds, and then we have got mammals as well, that is us. So this is the scale. So your fishes comes to be the second option. So the one that we have starting from second option is only these two. Let's cut down these two options. Eliminate them. Do not have confusions then. Then amphibians is 1. Amphibians is 1. And then we have reptiles 3. And then we have mammals 4. Then your answer will be C. Your answer would be C for this particular question. Okay? Okay. Next, antivenom given against snake poison contains what? Antivenom, my dear children. For example, I went to the doctor, a snake bit me, okay. I went to the doctor, doctor will give me something, which could save my life, right? And what is that something that could save my life? An antibody against this. An antibody against this. I cannot wait for my body to produce antibody against this. Remember always, the primary response is going to be slower. The secondary response would be good. But in the primary response itself, if I die, what's the use? So basically, doctor has to make sure that I gave the lady antibodies that she need that could resist the attack of what? The venom out there. So antivenom given against the snake poison contains not the antigens, not the antigen antibody complex, just the antibodies. You call it ready-made antibodies, or you also call it pre-formed antibodies, but you just give them. Similarly, in tetanus injection, what are you getting? Antitoxin is given to you. What is this antitoxin? The antibodies, so that your body can react, can fight back. Okay? You get it? You're giving everything out there. So for example, a soldier is there, a soldier is getting the gun and everything to protect himself. So this is the antibody that he has. If at all he does not have that, if he goes on searching that, my dear children, it will take a lot of time. Okay, and he might not be able to protect his life. Same thing happens over here. Please remember, this is a type of passive immunization. Your body is not producing antibodies, you have been given antibodies. So this is passive immunization. Active is when your body itself produces antibodies. Okay? So yes. Now, the substance produced by a cell in a viral infection that can protect other cells from further infection is a substance produced by a cell in a viral infection. So this particular cell, my dear children, is having viruses in it. And now, this cell, oh my God, what did I do? Okay, this cell which is there, it will be signaling other cells that, hey Chunnu, hey Munnu, hey, I don't know, people, you have to be protected. You have to be protected. For that, I am releasing something. So basically, they release interferons. And what do the interferons do? They go and act on the cell and tell that, hey, be ready, some virus attack is happening in my body, and it goes and tells the immunological cells to increase their number. And sometimes it makes the cell undergo something that is known as suicide, okay, cell death. Okay, cell death, you call it apoptosis. You don't need to remember it, I'm just telling you. That means if at all the virus comes and attacks, basically this particular area will be a place where the virus will replicate itself and increase its number, which is why the cells also undergo apoptosis, okay, so that they do not provide any area for the viruses to grow. So it is interferons which is being released, and it comes under cytokine barriers of innate immunity. It comes under cytokine barriers of innate immunity. I'm just making you remember things if at all you have forgotten somewhere, okay? Cool, ma'am. So because of the presence of which of the following vitamin, curd is considered as nutritionally richer than milk. Milk we get, curd we get from milk itself, then why is curd considered to be good a source of some vitamin, my dear children? Because, my dear children, milk, when it is converted to curd, it does not happen like that. You need the help of an bacteria that is known as lactic acid bacteria. So this lactic acid bacteria, my dear children, they release vitamin B12. They release vitamin B12 for their own functions for the cell to divide and the sunlight. But anyway they are releasing, and now it is there in the curd also. So curd is a rich source of vitamin B12. It is rich source of vitamin B12. Your answer would be this. Okay. Now mycorrhiza is involved in absorption of dash from the soil and passes it to the plant. So my dear children, I tell you one story. Okay, so there is a plant. This plant was very sad. Why? Because it was not getting any nutrition, and that's exactly when a fungi came into the picture. A fungus came into the picture. You know the name of this fungus, or basically where does it belong to? This fungus belongs to Glomus. This fungus belongs to a genus that is known as Glomus. And what does this fungus do? Basically, fungus, they have rhizoids which can go deep into the soil and can trap in the nutrients. Most importantly, it traps in a very important nutrient that is known as phosphorus, and gives it to the plant. He says that, hey, plant, you can take it. And now the plant is not sad anymore. He is happy, or basically she is happy. Whatever gender you want to take, you can take, okay? Why? Because it had got the enough phosphorus from the soil. I do not know why this goes into eraser mode automatically. There's some glitch, I suppose. Anyways, so it is phosphorus. Always remember, mycorrhiza is a combination of a fungi and the root of the plants. The fungus provides phosphorus. Apart from that, the fungus provides resistance to pathogen. Listen to me. Tolerance to salinity, if too much salt is there in the soil. Tolerance to drought condition. And not just that, my dear children, it is also responsible for one more thing I forgot. Yeah, for overall growth and development of the plant. It does not increase the yield. This is also a question. It does not increase the yield of the plant, does not increase the productivity of the plant. It's just that overall growth and development will also be increased. Now, what does the plant do for this? The plant basically provides organic nutrients because it is the one undergoing photosynthesis, right? That's why it provides the organic nutrients needed for the fungus. It's a symbiotic association where you are giving phosphorus. Okay, ma'am. Next question. In the given diagram of PBR 322 vector, identify the restriction site A, B, C, D. A, B, C, D. Okay. Now, this is this thing that you need to remember. How do you remember it? I'll tell you. Over here, every plasmid should be having origin of replication, ORI site. Then it has a very important thing which is ROP, a protein which is super important for the process of replication to happen. On these two sites, they have a restriction, they have ampicillin resistant site. That means this gene provides resistance to our antibiotic ampicillin. And over here, it provides resistance to tetracycline, which is another antibiotic out there. Now, my dear children, you have multiple restriction enzymes over here. The first one is this. In the ROP site, you have PVU2. PVU2. Okay. Here, you would be having two. One is BAMH1 and the other one is SAL1. Here, you have two again. One is PVU1 and one is PST1. Remember, PVU1 is here, PVU2 is here. So here you just have one wala thing, okay, two of them are there on either side. Here we have PVU2 and here we have three of them. So the first one that we have is the very famous EcoR1. The second then that we have is CLAV1 and the third one that we have is HIND2. Is what? HIND3. Okay, HIND3, not HIND2, it is HIND3. So 1, 2 and 3. 1, 2 and 3. 1 is over here and basically 2 over here and 3 over here. And remember, everyone has 1, 1, 1 except HIND3 and PVU2. Except HIND3 and PVU2. Now let's go into the diagram. So A is what? Two things are given. So A is the first one. It has to be origin of replication. It has to be origin of replication. So A is origin of replication. Then that's how I got my answer. B is what? Ampicillin resistant site. C is what? Tetracycline resistant site. What is the one line towards there? It is HIND3. It is HIND3. HIND2 was the first restriction enzyme. Okay, HIND2 was the first restriction enzyme. HIND3 is something different right over here. Okay, chalo. Go through the picture of this. Super important. Okay. Now, question number 12. The plants, don't think that this question would not be asked for you to draw. They might even ask you to draw. So please know this particular PBR 322. The plants developed by the fusion of protoplast of two different plants are called. So basically, I have a plant cell, another plant cell. You know what's the unique feature about plant cell? They have an extra layer that is known as cell wall. They have cell wall. Okay, suppose I am removing the cell wall from here. Now what I am left with? I am left with just the cell. You call this now as protoplast. What do you call this as? Protoplast. Suppose I am fusing the cells out there. What do you call this? Protoplasmic fusion. Or you call this as somatic hybridization. Okay, somatic hybridization. At the end of it, what are you getting? What do you call them? You call them as somaclons. No. Somatic hybrids. Oh my God. You call them as somatic hybrids. The one that undergoes somatic hybridization is known as somatic hybrids. Okay, somaclons are different. The tissue culture, in tissue culture from one part of the plant you get multiple plants being formed. They all look the same, which is why you call it as somaclons. So that happens in tissue culture, micropropagation, things like that. Here it is somatic hybrids. Okay, remember they undergo somatic hybridization and you are calling them somatic hybrids. Example, potato, tomato. I fuse them, I get tomato. Okay, so over here B is your answer for this question, somatic hybrids. Cool, ma'am. Let's go. Question number 13. In a pond, if there are 20 lotus plants last year, okay, through reproduction 16 are added, calculate the birth rate. Calculate the birth rate. So what is our initial population, my dear children? It was 20. What is the population, current population right now? It is what? 16. So basically we do 16 by 20 over here and we get the answer. So number of new individuals added. Okay, not the current population, rather we do number of new individuals added. Okay, number of new individuals added. Number of new individuals added plus, divided by what? Initial population into the time. It's just 1 year's time that has been mentioned over here. If yeah, because they have mentioned last year, right? That's why. Now 16 by 20 into 1 is equals to what? 0.8. So you should write 0.8 offsprings per lotus per year. Birth rate is mentioned like that. Per lotus per year. You should not write 0.8 offsprings per year. Okay, per lotus per year. That makes a huge lot of difference out there. So your answer for this question becomes 0. This. Okay, please remember the formula, might be needed. Okay. Now, the correct sequence of steps involved in decomposition is, what is the correct sequence of step involved in decomposition? First of all, suppose you have big things out there, you cannot directly ask decomposers to come in, that is fungi and bacteria to act on it. Rather, first we need to break it down, and that is being done by detritivores. Detritivores first, such as earthworm and things like that, they make the thing undergo fragmentation. Now we have broken down the things, and when we break down the things, my dear children, obviously there will be certain water soluble content which can quickly go seep into the soil. The water soluble content will go seep into the soil, and this process is known as leaching. Now whatever is left behind, okay, it is being acted upon by the decomposers. And now the decomposers such as bacteria and fungi, or F and B, that is I remember as Facebook, fungi and bacteria, decomposers would be starting acting on this, and this process of beginning up the process of decomposition, of breaking down the organic matter, okay, or breaking down the organic matter to inorganic matter is known as catabolism. But it is not a one-step process. It is not a one-stop process. Whenever you make things undergo catabolism, what is going to happen? Basically, we get so from organic to inorganic, it's not a smooth journey. Suppose this is organic, suppose this is inorganic. When you reach halfway, okay, you say it is partially digested. It is what? Partially digested. And in this condition, you call this as humus. You call all the organic content as what? Humus. It is not fully been digested. Okay, it is not fully been digested. You call this as humus, and this process is known as humification. Listen, it is highly rich in all the nutrients because you're trying to break it down slowly, bit by bit. So it is partially digested form. It is highly rich in nutrients, and it gives a dark amorphous color. It gives a dark amorphous color to the soil out there. Now later, this whole process is being completed. The second half will be known as mineralization, where finally it is fully broken down, and you get all the inorganic nutrients. So always understand, catabolism itself can be divided into humification as well as what you call mineralization. So the children usually think this is a whole different process, this is a whole different process, this is a whole different process. No. Catabolism means organic, okay, complex organic component into simpler inorganic components out there. And while they do it, you get humus, and this process is known as humification. Later on, again it is broken down. You call this as mineralization, where finally the inorganic nutrients are gotten back into the soil. So your answer for this question would be A, fragmentation, then leaching, then catabolism, then humification and mineralization. Cool? Are you understanding what I'm trying to say, or is it difficult for you to understand? Well, I hope it is easy. I'm trying to spend my time on questions and make you understand. Well, my dear children, if at all you're not understanding, please let me know, okay? And if at all you're understanding, if you think the teaching is good, then let me know that as well. Okay, question number 15. This is the last MCQ question, then we will be having fill in the blanks question. The following are the in situ conservation approaches except. In situ means you are conserving organisms in their natural habitat. In their natural habitat. So there's a question that the following are in situ conservation except. So you have to tell what is the exception, which one is not an in situ form of conservation. National parks it is, wildlife sanctuaries it is, biosphere reserves it is, botanical gardens it is not. You might have seen Mysore Botanical Garden, my dear children, all the here Lal Bagh Botanical Garden in Bangalore, and wherever you're staying, you might have seen botanical gardens. There, you take the plant and you grow it elsewhere. It is ex situ conservation, not within their actual area, my dear children. So this is an exception. Okay, this is the exception out there. Now remember, wildlife sanctuary and wildlife safari parks are different. Wildlife safari park is ex situ. Wildlife sanctuary is in situ. Always have that in your head. Sometimes children get it wrong. Okay, so wildlife park is different, wildlife sanctuary is different. Now we go ahead with what? The fill in the blanks question. Now this is the place where I feel so happy because a very easy to score place is this, right? So the glandular layer of uterus that undergoes cyclic changes during menstrual cycle is the glandular layer, okay, the one that secretes things, okay, the one that undergoes cyclic changes every now and then. It is shut down and grown, made up again. What do you call this layer as? This is the innermost layer of uterus. You call this as endometrium. You call this as endometrium. So for 16, your answer is endometrium. It is not myometrium. Okay, uterus has 3 layers. The outer one is perimetrium. Middle one is myometrium. It is made up of muscles, smooth muscles, involuntary muscles. Smooth muscles are always involuntary. You cannot control them. Your brain does. Okay, and then we have the innermost one, which is endometrium. Next, the paleontological evidence for evolution is obtained from. Paleontological evidence that evolution has happened. Where do you get it from? Basically, have you watched Friends? In that Ross works as a paleontologist, right? So basically it's fossils. It's fossils that gives us information that there is an evolution that has taken place, and these fossils, when we say that fossil is the evidence, you call it as a paleontological evidence. So 17th answer is fossils. Now 18, dash is a very effective sedative and a painkiller. Sedative means you feel to sleep, and it's also a painkiller. So doctors usually after surgeries, they give this drug to the patients because those of you who cannot bear the pain at all, it is being given. Otherwise you should not be taking in. This particular drug is an opium. Okay, this is an opioid, my dear children. And what is the answer for this question? It is morphine. It is morphine. Morphine, when it undergoes a process of acetylation, you get diacetyl morphine, that is heroin. 2 different drugs, both of them coming under opioids. This one is a sedative and a painkiller. Okay, now the plasmid DNA act as dash to transfer the piece of DNA attached to it. So the plasmid DNA is there, and this plasmid DNA, my dear children, understand what is a plasmid and things like that. I have taught you in Brahmastra series. My dear children, by the way, if at all you are facing any trouble while solving questions of a chapter, please watch the Brahmastra series. I am telling you, it's super helpful. We are giving it for free. Whenever we give things for free, you would not watch it. I do not understand the logic. So whenever there is a price tag being attached, you guys are like all crazy. But why not use the free resources as well when we are putting in lots of efforts for that? So please do that. It's really, really helpful for you. So previous year, lots of children took the benefit of it. So please do that. So the plasmid DNA act as dash to transfer the piece of DNA. So this is the piece of DNA we want, and plasmid DNA is going to act like a vector. It is going to act like a vector to help it carry. So over here, your answer for this question becomes vector. Now, the genetic diversity shown by Rauwolfia vomitoria growing in Himalayan ranges produces an active chemical known as. The genetic diversity shown by Rauwolfia vomitoria. What does it produce, my dear children? It produces something that is known as reserpine. Okay, what do you call this? This is known as reserpine. Why do you think it is being produced? Basically, it slows down our nervous system. It slows down the BP, blood pressure and everything out there. So it is reserpine that you need to write the answer as. Okay, these are the 20 questions. Now going ahead with the next question, 21st question. Write the significance of sporopollen. From here we are entering the 2 marker zone. So for those of you who have, just a second, okay? Just a second, quick second. Okay, so those of you who have reached until here, comment down in the comment section that, ma'am, we are done with the first part, that is the MCQs or basically the 1 marker session. I really want to know who all have reached until this particular point. Okay. Now, so our 30 minutes, if at all it is being finished, pause the video, try to recollect all the questions out there, try to solve all the MCQs once more and see how far have you come. And my dear children, post that, you can restart this video, okay? So what is the significance of sporopollen? That is the first question. What is sporopollen? My dear children, you might have seen pollen grain. The pollen grain, my dear children, has a plasma membrane for its own. Apart from that, it has another layer which is known as intine, and then it has one more layer which is discontinuous. Okay, it is discontinuous. It have pores like this. You call this as exine. What is this layer made up of? It is made up of sporopollenin. Sporopollenin is the toughest organic matter ever known. It can withstand high alkali, acids, anything out there. And which is why it can protect the plant pollen grains, and the pollen grains can be preserved as fossils as well. That's the power of sporopollenin. The intine, on the other hand, is made up of Priyanka Chopra. You can remember like that: P for pectin and C for cellulose. Okay, or PC, the PC that you have computers, you can remember like that also. Write the significance of sporopollen. It can sustain strong acids and alkalis, as it is one of the most resistant organic substances ever known. It protects the pollen grains from physical and biological damage, helping them to survive under adverse conditions. They can also be stored as fossils. They can also be stored as fossils. Please remember that. And the area where the sporopollen is not there, you have a pore. You call this as germ pore. You call this as germ pore. In the model question paper 1, we solved the question where basically they are asked what is inside the cell. Please go through all those questions. All the model question papers, solve them. I'm not saying the questions would be coming exactly like that, but I'm giving you an idea that the concepts, the repeated concepts would be put down there. You will have a good, practice of questions, okay, of the concepts in that way. Now mention the reasons for infertility in humans. Why do you think human beings are becoming infertile, or why there is infertility in the case of humans? Why are they not being able to give rise to a baby? Okay, why? So first thing, physical. There might be physical reasons because of their body's problems. There might be congenital reasons, as in something that is genetic, that they inherit, my dear children. There can be disease-related causes as well. Okay, so structural defects in the reproductive organs comes under what? Physical thing. Hormonal imbalance and everything also comes under physical. Now you have infections, diseases. Sorry, hormonal imbalance can be coming under congenital as well. So suppose my mother has a thyroid deficiency, and if the same thing happens to me in some other way, probably it could be related to some inherited thing. And if thyroid deficiency is there, obviously it would be hampering the fertility as well. Okay, now disease-related causes such as infection is there, or some disease. You have some sexually transmitted disease that is preventing from fertilization to happen. All of this comes under this. So 1, 2, 3, write your answer like that. I just gave you a gist. So for physical, you write this. For congenital, you can write like that. For disease-related, you can write this. Okay. Then you have drug-related. People take drugs, and these drugs, my dear children, have some effects on our body, obviously. So that could be a reason. Immunological reasons could be there. For example, our immune system might be acting against gamete. You might have seen autoimmune disorders, right? Where our own cells are being killed. This is known as autoimmune disorders. So immunological reasons could be there. Apart from that, psychological causes. Yes, stress and everything obviously leads to what? Hormonal imbalance out there, leading to infertility. So these are the reasons for infertility in the case of humans. Please remember them. Don't mug it up. Try to understand. Okay. Okay, ma'am. Now question number 23, differentiate between linkage and recombination. Linkage and recombination, what do you mean by that? I'll give you an example. Don't read this. First, let me give you the example. So over here, I have a chromosome that looks like this, with capital A gene and capital B gene over here, and a small a gene and small b gene over here. I have another one where I have capital A, capital B right over here, and small a, small b gene right over here. Now, my dear children, When these chromosomes are lying close to each other, there is a process that usually happens, okay, in the case of human beings and all the organisms that undergo sexual reproduction and things like that, and that is known as recombination. That is known as crossing over. What do you call it as? Crossing over. So in crossing over, suppose this part of the chromosome went here, this part of the chromosome went here. What do you get now? You get a chromosome that looks like this. So initially capital A was with capital B, but over here small a is with capital B. You get it? There is a new combinations being formed. There is a new combination being formed over here. Right? Now if at all you see this condition, now from here to here if it is mixed, capital A, capital B was on this side, now it will be on that side. And this thing came this side. But anyway, these genes are what? Still together. So amount of recombination which is there, recombination is happening obviously. The upper part of the things has been changed. But among them, the recombination is what? Very much less. Among these genes, the recombination would be very much less. So over here, you need to understand that these genes which are there, you call them as loosely linked genes. And loosely linked genes, they show high amount of recombination. On the other hand, you have other genes right there, you call it tightly linked genes. You call them tightly linked genes. And what do? They basically show lesser amount of recombinations out there. Okay, so linkage is what? It is a tendency of genes located close together on the same chromosome to be inherited together. It's a tendency of the genes which are close together to be inherited together. Whereas recombination is the process of creating new combinations of genes formed due to what? Crossing over. Now linkage, it reduces the chance of new gene combinations and maintains the parental character. Whichever parental character was there, that is being maintained. But over here, recombination, it increases genetic variations, my dear children, producing new gene combination. So one focuses on parental characteristic, whereas the other produces new gene combination. Who put up these two terms? It was Thomas Hunt Morgan. It was Thomas Hunt Morgan who gave linkage and recombination. And with that, his student Alfred Sturtevant was able to come up with something else that is known as gene mapping. It is known as gene mapping. So he said that, okay, if at all I know the percentage of recombination, if percentage of recombination is this much, 37%, that means there is too much amount of gap between the genes. And that's how I can locate the genes on the chromosome as well. It is known as gene mapping. It is known as gene mapping. If at all recombination is less, that means probably they are close to each other. And I'll get to know that this gene is located over here, the other gene is located right over here. I get it? So that is the difference between linkage and recombination. Next question, draw a label diagram of nucleosome. So this is a nucleosome. Your DNA cannot fit inside the nucleus easily. Your nucleus is this much, your DNA is very long, 6.6 into 10 to the power 9 base pairs. So how do we fit it in? So basically we have the help of some proteins out there. Okay, you call these proteins as histone proteins. Histone proteins. They are almost 8 in number. How many? 8 in number, which is why I'll call this histone octamer. On this histone protein, the DNA is going to be wrapped around. How many times? Twice. How many times? Just twice. How many base pairs of DNA will be there? Probably almost 200 base pairs of DNA is going to be there. Why does the DNA wrap around this histone protein? Because, my dear children, DNA which is there, it is negatively charged, whereas the histone protein which is there, it is positively charged. Why is it positive, ma'am? Because it is made up of amino acids which are arginine and lysine. Arginine and lysine, both of them are positively charged amino acid. Always remember it. Okay, now apart from these histone proteins, you have one more histone protein over here. Okay, right over here, you call this histone protein as H1 histone. You call this as H1 histone. Now the same thing is going to be repeated again. Here you have the histone protein, H1 histone. Okay, there can be long stretches where there is so much gap, you call it euchromatin. Okay, anyway, first of all, when you have a bead on a string-like structure, you call it as chromatin. Chromatin is of two different types. If at all you have lots of space, you call it euchromatin. If they are very much cramped, you call it heterochromatin. There's a question being asked about what is the difference between euchromatin and heterochromatin. Go through it as well. Okay, next question. Comment on the similarity between thorn of bougainvillea and tendril of cucurbita with reference to evolution. So thorn of bougainvillea. and tendril of cucurbita, my dear children, both of them arise from axillary bud. So in bougainvillea it becomes a thorn. In the case of cucurbita it becomes a tendril. But both of them are arising from the same structure, different function. Okay. Arising from the same structure but having different function. Why do you think they have different function? Because they are in two different habitats. One place needs protection, other place needs support. So this is that basically they evolved from the same position, but they moved to different things. Okay, it is example of divergent evolution. It is example of divergent evolution. And these organs that you see, what do you call them? The organs that show divergent evolution, you call them homologous organs. You call them homologous organs. Okay, you can remember HD, homologous organs, divergent evolution. So similarity between thorn of bougainvillea and tendril of cucurbita is that the thorn of bougainvillea are homologous organs. Both originate from the same basic structure, that is what, but does not have the same basic structure. Okay, please understand, they originate from the same basic structure, that is your axillary bud. Now, but perform different function, originating from the same but performing different function. Thorn, protection, tendril, climbing. Evolutionary significance is that this similarity indicates divergent evolution, where structures with common origin evolve to perform different functions due to what? Different adaptations. So that is it. You have to mention homologous organs, underline that. You have to mention divergent evolution, underline that also properly. Cool? Yes, ma'am. Now, what are flocks? Write their significance in sewage treatment. Now, flocks and BOD is something very, very important for you. What is flocks? So, my dear children, usually when you do sewage treatment, there is a primary treatment being done, where whatever thing is there, it is put in a settling tank to settle down. So, whatever thing sediments down, you remove it through a process of sedimentation. And whatever things floats around, you remove it through filtration. You remove it to filtration, and whatever is left behind, you call this as a primary effluent. Primary effluent. This primary effluent is taken to the next tank, which is known as aeration tank. For the next step, that is secondary treatment. Now, in aeration tank, my dear children, okay, whatever organic matter is there in this primary effluent, that has to be treated by some aerobic bacteria. That is going to be treated by some aerobic bacteria. And they do not do it alone. They take the help of some fungal filaments. So together, the aerobic bacteria plus the fungal filament, you will now call this as flocks. Now this flocks is going to act on the organic matter and break it down in the presence of oxygen. Remember, it's aerobic bacteria out there. They will work until the level of organic matter is reduced. That means the pollution is reduced. And my dear children, with that I'll also talk about BOD, biochemical oxygen demand. My dear children, this aerobic bacteria which is up there, it definitely needs oxygen to break the organic matter. So if at all organic matter is more, aerobic bacteria will be more, they will also need oxygen more. That means biochemical oxygen demand is more. There is more demand for oxygen. Whereas if at all organic matter is less, that means pollution is less. The aerobic bacteria will be less. Okay, oxygen needed will also be what? Less. That means biochemical oxygen demand will be less. So we work until the biochemical oxygen demand goes low. That's when we understand that pollution is less. So flocks are loose aggregates of aerobic microorganisms, mainly bacteria along with fungal filaments. What bacteria? Please write. It is aerobic bacteria. I had missed it. Formed in the aeration tank during the secondary sewage treatment. Significance of flocks in the sewage treatment. They break down the organic matter present in the sewage. This reduces BOD, biochemical oxygen demand. Lower BOD means reduced pollution potential to the effluent. They help in purifying sewage biologically before it is being released. Got it? Yes, ma'am. So flocks and this super important. Everything of sewage treatment is actually important. Go through it. Now mention the evil quartet of biodiversity losses in a given habitat. So there are 4 reasons, major reasons why biodiversity loss is happening. And you call it evil quartet. Quartet means 4. First one is habitat loss of fragmentation. Suppose organism is living there, we do deforestation out there. Where will the organism go and live? Obviously they will be lost. Biodiversity loss is happening out there, or basically we cut the land into two and divide them. Obviously they do not have places to move around, and that's how we lose things, right? So one is this, overexploitation. Okay, sometimes we go on hunting things. Okay, we hunt, and what happens, my dear children? The organism, it becomes extinct. So this is overexploitation. Alien species invasion. Sometimes, for example, Ecornia, Parthenium, it was exported from outside because of its beauty. But what does Ecornia do in the land, my dear children? Basically, in the water, they make sure that none of the other plants are getting enough sunlight, they are not having any source of oxygen and things like that. Because first of all, Ecornia spreads through, so sunlight does not reach. Then when the Ecornia die, they basically are acted upon by bacteria which needs oxygen, depleting the level of oxygen out there. And when oxygen is not there, obviously other organisms cannot also not survive. This is an example. You have multiple examples out there. Now co-extinction, there might be some organisms which says that some flower out there which is pollinated by certain groups of plant. For example, you have the wasp and the fig tree. Okay, both of them show mutualism. If one organism gets extinct, what will happen? The other organism will automatically go extinct. So this is what co-extinction. So you have 4 reasons: habitat loss and fragmentation, overexploitation, alien species invasion, and co-extinctions. Done and done. And now we move on to the 3 marker questions out there. Draw a label diagram of LS of an embryo of a grass. What do you mean by grass? It's a monocot or is it a dicot? Grass, my dear children, it is a monocot. So basically you are being asked, draw the diagram or draw the LS of a monocot. So how do you do it? Monocot means they just have one cotyledon. My dear children, understand, you have an embryonal axis up there, and this embryonal axis, my dear children, would be having one cotyledon. What do you call this cotyledon as? You call this cotyledon as scutellum. Embryonal axis means an axis which will be giving rise to plumule. Plumule means the shoot tip. Okay, it gives rise to the shoot. And then you have radicle. The radicle is the root tip. The radicle is always covered by something that is known as the root cap. The root cap. And over here, monocots, why do you think they just have one cotyledon? Because they were not able to utilize all the endosperm. So I'll tell you one very important thing. You have endosperm, you have zygote. Endosperm is something that helps the zygote transform into an embryo. Now when an organism becomes an embryo now, so it has taken up the endosperm already. Okay, and this embryo has its own form of storage. You call it as cotyledon. But some type of embryo, they do not use the entire endosperm. In that case, you have little endosperm still being left around. And that happens in the case of monocots. Dicots do not have it. Dicots basically have 2 cotyledon. So whatever endosperm content was there, they made into cotyledons and they are storing it. Monocots are not like that. For example, grass, wheat, maize, barley, all of them, they just have one cotyledon, and remaining is the endosperm out there. Okay. Now the plumule which is there, it is covered by something which you call as coleoptile. PP, you can remember like that. And the root cap over here, it is covered by something that is known as coleorhiza. RR, you can remember like that. Now this is the actual picture. We have to come to this. So for that you need to look at the picture once more. Plumule covered by coleoptile, radicle covered by root cap coleorhiza, scutellum on one side, and then endosperm. Look at this picture. Shoot apex as in your plumule covered by coleoptile, then you have radicle, root cap and coleorhiza. You have the one cotyledon that is known as scutellum. The other cotyledon tries to be formed, but it is rudimentary. It is not able to fully form. It is known as epiblast. Now how do you come to make this particular diagram over here? That is kind of tricky, right? So what do? We try to make it. So remember, it's a fish's mouth. Okay, shark is there, and the fish has a hand out there, and then we go to the tail. So this is the tail, and then a small fin right over here. This is the small fin. Now my dear children, make a V like this. Okay, make another V like this. Cool. Now join this. Let's go here. Let it come back, go to one tail, and let it join back right over here. Over here, make one V, and that's all. So this is what? This would be your coleoptile. This is going to be your plumule. This is going to be your scutellum. This is going to be your radicle. This is your root cap. This is going to be your coleorhiza. And this is going to be your epiblast. And we are done with the diagram. Okay, make this, it will be more than enough. Trust me, forget stains. Yes. Now, next question. What is placenta? List its function. What do you think is placenta, my dear children? So basically, this is the uterus, right? Now the baby comes, the baby does not form directly. You have the embryo being formed. There is one stage of the embryo, you call this stage as blastocyst stage. I'll tell you a unique feature about blastocyst stage. What is a unique feature about the blastocyst stage? It has the outer layer of cells, which you call as trophoblast, and the inner layer of cells right over here, you call it as inner cell mass, right? Now this blastocyst stage, when it reaches over here, they slowly get embedded into the uterus. They slowly get embedded into the uterus. Now the trophoblast which is there, the outer layer which is there, it forms finger-like projections, okay? It forms finger-like projections inside the uterus, inside the walls of the uterus. You call this finger-like projections as what? Chorionic villi. Chorionic villi. From the maternal side, from the mother's tissue side also, they form some blood spaces around it. They form some structures around it. Together, both of it, the blood spaces, the blood-filled spaces as well as some part of the tissues, my dear children, some part of the maternal tissue, together it now forms a structure that is known as placenta. It forms a structure that is known as placenta. So if at all we see now, there will be, where is it? There will be placenta, and attached to it, with the help of only trophoblast helps in chorionic villi formation, right? The inner cell mass is still left. The inner cell mass is now going to give rise to what? The whole embryo out there. The whole embryo out there. And both of them are attached to a cord which is known as umbilical cord. Umbilical cord. Okay, so what is placenta? It's a vascular connection. It's a vascular connection between the mother and the fetus that helps in nutrition, respiration, excretion and hormone secretion. Nutrition how? Transfers nutrients from the mother to the fetus. Okay, respiration how? Supplies oxygen to the fetus and removes what? Carbon dioxide. Excretion removes metabolic waste from the fetal blood. Endocrine function. This is one of the most important thing. They release 4 types of hormones. Remember, it's a temporary endocrine tissue. Okay, temporary endocrine gland out there. Only during pregnancy it will be there. What does it release? HCG, human chorionic gonadotrophin. HPL, human placental lactogen. Progesterone, estrogens, all of them are released by placenta. Please remember 4 hormones. Only 4 hormones are released by placenta, and those are this. So they were initially released by the ovary and things like that, but during pregnancy they take it over. Okay, so HCG, HPL, progesterone and estrogen are some things released by the placenta. HCG is super important, okay, for maintaining corpus luteum. Corpus luteum will keep on producing progesterone, and that's how it works. So, remember luteinizing hormone LH was there in menstrual cycle, which was helping the corpus luteum to be maintained, and this corpus luteum was releasing progesterone. But now, if at all, you need someone to be there to keep the corpus luteum to be ready to still produce progesterone. You need someone, and that is HCG. HCG helps in corpus helps in the survival of corpus luteum, and which is why you get progesterone. Now, if at all this point, if you feel it's very difficult for you to understand, chill. Remember this, okay? That's all. Now, define venereal disease or sexually transmitted infections. Mention the modes of transmission of STIs. Venereal or sexually transmitted diseases are infections that are primarily spread through sexual contact with the infected person. Okay? Example, you have to talk about the examples. I have not mentioned HIV, AIDS, herpes, gonorrhea, syphilis. Please write the examples as well. Now, modes of transmission. Unprotected sexual intercourse is the first one. Without condoms, sexual intercourse you might get because it is passed through the body fluids out there. Second, sharing contaminated needles or syringes. Listen, one very important thing, not all of the sexually transmitted diseases are transferred like this also. You have exceptions like HIV is there, which also spreads to the other body fluids like blood. Okay, so which is why I'm saying that modes of transmission of STI is not just limited to unprotected sexual intercourse. For some diseases, it can be spread through sharing contaminated needles or syringes because blood is being involved there. Transfusion of infected blood or the blood products. For example, a person who undergoes repeated blood transfusion, a person who has thalassemia has to undergo repeated blood transfusion. So in that particular case, my dear children, if the blood is not being checked properly, if the blood is infected, this person will also get the infection. Now, from infected mother to the fetus during pregnancy. During pregnancy, if there are slight chances that from the infected mother it can pass to the fetus, even though now you have different ways to prevent that, but still, okay, it can happen even during breastfeeding. So these are the modes of transmission of STIs. Remember, primarily it is unprotected sexual intercourse. There are some exceptions to it, some diseases which can be spread through other body fluids, and that has been mentioned over here. Okay, the ways. Cool, cool, ma'am. Now draw a label diagram of Miller's experimental setup. Now I won't be drawing that, but I can definitely show you. So over here in the Stanley Miller's experiment, he said that chemical evolution is happening. What is chemical evolution? That basically in the previous earth that we had, or primitive earth, not previous, primitive earth that we had, there was no atmosphere. There were some gases out there such as methane, such as ammonia, such as H2 in vapor format, and H2, hydrogen gas. And the temperature was super high. Okay, temperature was super high and there was no oxygen. So there was no oxygen. So we have to maintain all this condition over here. So let's do that. So this is a closed setup over here. You have water. The boiling water is there. The vapors are going to grow up there. You have electrodes. Why do you have electrodes? To increase the temperature. The temperature over here in Stanley Miller's experiment is 800 degrees Celsius. Remember this. It is 800 degrees Celsius. Now, when this thing is there and when you have all the gases combined together with the inorganic things that you are having over here, towards this particular place where everything, the gases are going to settle down in the liquid water, you get organic content. You get organic content out there. And was the first organic content that was found by Miller, it was amino acids. It was what? Amino acids. Nothing else but amino acids. So draw a label diagram of Miller's experimental setup. This is the thing. Please go through it. Super important, especially these gases. Methane is not CH3, it is CH4, and NH3, I also used to do this mistake, that's why I'm telling you. NH3 is ammonia, H2O, and H2. Because, well, you are very prone to studying chemistry, so you might know, but still, there are chances we all do mistakes, right, in life. Next, mention the causative organisms for the following diseases: Escherichiasis, typhoid, filariasis. Escherichiasis, typhoid, filariasis, the causative organism. Escherichiasis is caused by Ascaris lumbricoides. It's a roundworm, Aschelminthes. Okay, it's a roundworm. Typhoid is caused by a bacteria, my dear children. What do you call it? Salmonella typhi. Salmonella typhi. Now, filariasis is caused by another Aschelminthes. It is known as Wuchereria bancrofti. Wuchereria bancrofti, you have another name for it, another species for it, and that is Wuchereria malai. Okay, Wuchereria malai. So here I have all the things written properly so that you do not need to worry. Remember, malaria. Why is malaria mentioned over here? I think I put down the wrong answer. Yes. So I put down the wrong answer, but still you can go through this as well. So this is totally different disease, but anyways. So Escherichiasis is caused by Ascaris. Typhoid is caused by Salmonella typhi. So you might be thinking, ma'am knows how to write properly also, right? Then why does she not do? So Wuchereria is written over here. Do you want me to write? I can write it. So you have Wuchereria bancrofti, and you have Wuchereria malai. Okay, fine. So this is a bacterium. These two are Ascalvinthus. You have ringworm caused by fungus, that is trichophyton, epidermophyton, and all of them, and microsporum. Yes. Malaria is caused by Plasmodium vivax, Plasmodium falciparum, Plasmodium malariae. Plasmodium falciparum causes the malignant form of malaria. Extra question, extra answer. So yes. Now name the microbes that help in the production of the following bioactive agents. Bioactive agents, this is from the chapter Microbes in Human Welfare. So cyclosporin A, what do you think it is? Cyclosporin A is an immunosuppressive agent. That means, for example, my kidney is not working well. I have taken the kidney from someone else, transplanted the kidney. Now my body feels something foreign has come inside. I have to kill it because it could be dangerous. But it's something for me. My body does not know it. I cannot not tell it to my cells, right? So basically, our body is given immunosuppressive agents that suppresses down the immune system a bit, and that helps in organ transplantation process. So cyclosporin A is that immunosuppressive agent, and you get it from Trichoderma polysporum. You get it from Trichoderma polysporum, which is a fungi. Which is what? A fungi. It's a fun guy. You can remember like that. Cyclosporin A from Trichoderma polysporum, and it's a fungi. Now, streptokinase. Streptokinase is a clot buster. You might have seen people having heart attack, myocardial infarction, you call it in scientific terms. Why do they have it? Because there might be blockage in the arteries which carries the blood, my dear children. There might be bad cholesterol being formed up there. So you need clot busters to be given. And streptokinase is one such enzyme. It's an enzyme, and it's a clot buster. Where do you get it from? You get it from Streptococcus, that's a bacteria. So it's a clot buster to dissolve blood clots in patients with myocardial infarction or heart attack, and you get it from Streptococcus, that is a bacteria. So this question is about, tell the names of the microbes. So, and what are they? So basically write about bacteria also, that they are bacteria, that they are fungi and everything. Now we have statin. Statin is for stabilizing blood cholesterol level. I remember like that. It is actually for lowering down the blood cholesterol level, but I remember stabilizing blood cholesterol level. So please remember both stabilizing and lowering the blood cholesterol level, and you get it from Monascus purpureus. I know the names are not so fascinating. It is Monascus purpureus. It's also a fun guy. Okay, it's also a fun guy, and as I told you, it's a blood cholesterol lowering agent. Okay, so these are the bioactive agents. Please remember them. These are repeated so many times in in the other model question paper also. You could have seen that what are the functions is being asked. So please know about them. Now schematically represent pyramid of energy with appropriate units. So how do you show pyramid of energy? It cannot be inverted. It will be always upright like this. So my dear children, we get almost around, suppose we get 10 lakh joules of sunlight. We get 10 lakh joules of sunlight. Out of that, only 1% of it, if we get, if we get 1% of it, usually from the sunlight that we have, my dear children, plant does not take everything. Just 1 or 2% has been taken. So if at all I take 1%, that means 1 into 100. So 100, 0 cancels over here. I just have 10,000 joules in the plant. Now the plant which has 10,000 joules, do you think it will give everything to the next organism that eats it? For example, a rabbit eats it. So over here I have written primary producer. Okay, this is the primary producer, this is the primary consumer, secondary consumer, tertiary consumer. Don't put shortcuts like this, write entirely primary producer. So what is it? It's a plant. Suppose the plant is being eaten up by rabbit, and rabbit is eaten up by snake. Okay, rabbit is eaten up by snake, and snake is eaten up by hawk or anything out there. Okay, or probably, yeah, that also works. So my dear children, you need to understand that you need to write everything trophic level 1, trophic level 2, trophic level 3, like that, and over here mention that energy in joules, my dear children, with appropriate units being written over here. Now 10,000 amount of joules which it has, do you think everything is passed to the next one? No. According to 10% law, only 10% of the things are going to be passed. Only 10% of it is going to be passed. So what is 10% of it, my dear children? It will be 1,000 joules. Now again 10%, now again 10%. What about the 90%, ma'am? 90% of energy will be used by the organism also. No, it has to respirate, it has to reproduce, lot of other functions are there. So that is why 10% law is there. the 10% law states that only 10% of the energy available at one trophic level is transferred to the next higher trophic level, while the rest is lost as heat and metabolic activities. Got it? Yes, ma'am. So make this and your life will be jingalala. And now we have reached the 5 mark question era. And with that we have the first question. Explain the different outbreeding devices developed by flowering plants. Before that, let's see how many questions are left. Few. Very few. Only this much is left. Okay. Only this much is left. Please do not be worried. We will do like this. Chalo. So different outbreeding devices developed by flowering plants. Outbreeding means basically I do not want the plant to undergo self-pollination. I do not want the plant to undergo self-pollination. Suppose a plant has the female and the male part over here. I have to prevent it to undergo self-pollination. Why? Because it can lead to inbreeding depression. The same genetic things are being passed on again and again. Suppose this genetic information is not that great. The plant is not that great. Then progenies will not be able to survive, right? That's why we promote outbreeding devices, not we, the plant. For that, the first thing is time difference. Pollen release and stigma receptivity not synchronized. The pollen is released, but the stigma is not receptive. In that case, obviously, pollination is not going to happen from somewhere else. The pollen might be coming and landing up there, right? So this is one thing. Second thing, spatial separation. We have what? Spatial separation. That means anther and the stigma are located at different positions. For example, your anther is up over here and stigma is up over there. Stigma is up over here and anther is up over here. So this is what? Spatial separation. Anther and stigma are positioned differently. Now from here to here it cannot flow and go, fly and go, right? So that is one thing. Self-incompatibility, that is a genetic mechanism. Self-incompatibility, my dear children, it's a genetic mechanism that prevents self-pollen from fertilizing the ovules. Suppose over here it landed on the stigma, everything is receptive also, but what happened? Because of some chemical blockage out there, it is not able to form the pollen tube. This is known as self-incompatibility. Okay, your own pollen grains cannot undergo the formation of pollen tube and things like that. So genetic mechanism prevents self-pollen from fertilizing the ovules. This is self-incompatibility. Now we have formation of unisexual flowers. That is the next thing. Formation of what? Unisexual flowers. How does it help, ma'am? How does it help? So I'll tell you. Listen to me carefully, okay? So for that I need to go something like this. So over here I have a plant, another plant. Okay? This is papaya plant. This is what? Papaya or watermelon. Papaya or watermelon. Anyways, so over here I have the female flowers. And over here I have the male flowers. From here to here, do you think pollination can happen? From here to here, do you think pollination can happen? No, ma'am, it cannot. That means forcibly from here to here it should be happening. Cross pollination has to happen. Yes, these are dioecious plants. What do you call them as? You call them as dioecious plants. Here, the sexes are separate. You will be having a female plant, not a female flower, female plant and a male plant. So these are dioecious plants. Now you have something that is known as monoecious plants. So monoecious, my dear children, are very unique. Monoecious such as castor and everything. Okay, castor is what? Monoecious. What is exactly happening over here? One plant will have both the sexes. Now, there can be two conditions. One condition is where it's like this, or probably there can be a condition where it can be like this. A flower having both the sexes, or a plant having flowers, two separate flowers, male flower as well as the female flowers. These are monoecious plants. They are what? Monoecious plants. So in monoecious plants you have different types. I'm talking about this one. here to some extent, my dear children, I can prevent self-pollination to some extent. How? Autogamy is not going to happen. Autogamy means from the same flower to the same this is known as autogamy. This would be prevented over here. This would be prevented over here. Geitonogamy can still happen. Geitonogamy means between the flowers of the same plant. From here to here it can happen. So geitonogamy is happening, but autogamy is prevented here. Autogamy is prevented. Geitonogamy is still happening. Geitonogamy is still happening. And cross-pollination can happen. Xenogamy can also happen over here. But autogamy is prevented in this condition. If unisexual flowers are produced on a bisexual plant. Okay, unisexual flowers are produced on a bisexual plant, you can prevent this. Otherwise, you have a better option, that is dioecious plants. Got it? Take a screenshot. It is definitely going to help you out. So they will ask you what is happening over here. Here autogamy cannot happen definitely, and geitonogamy is also prevented. Geitonogamy is also prevented. Only thing you have is xenogamy. Only thing is xenogamy. Here autogamy is prevented again, but geitonogamy is not prevented. And xenogamy can also happen over here. You get it? So please take this down. Very, very important. So unisexual flowers, monoecious, example castor and maize. Male and female flowers on the same plant prevents autogamy, not geitonogamy. Not all monoecious plants are like castor and maize. Please remember that as well. Now dioecious plants, papaya, male and female flowers on different plants prevents both autogamy as well as geitonogamy. This is papaya male, this is papaya female plant. And this is where you have male and female plant together. This is castor. You get castor oil from castor. This is the castor. Okay? Now, draw a neat label diagram of the sectional view of the mammary gland. So again, over here, I can help you with how you can remember things. So, dal fell on mat. Dal fell on mat. What is M over here? It is mammary lobes. Inside the mammary lobes, what do you have? You have mammary alveoli. Inside that, the alveoli basically comes together to form what? The mammary tubules. The mammary tubules comes together to form what? The duct. The duct comes together to form what? The ampulla. And the ampulla comes together to form the lactiferous ducts. Lactiferous ducts. So ma'am, how do I remember alveoli and ampulla? Ampulla ends with L, so the next letter is L, and that is what? Lactiferous duct. Dal fell on mat, or mat is there on dal. Either way, this will be super enough for you to remember things, the order at least. Now see the picture. These are some mammary lobes. Okay, they are what? The mammary lobes. Inside that you have the alveoli. Alveoli, my dear children, if at all you see this gap, one tubule, second tubule. Okay, one tubule, second tubule. Tubule is not being mentioned over here, but you should know. Tubules come together to form the duct. So this is the duct. This thing that you see is what? A duct. Ducts come together to form what? The ampulla. This is the ampulla. The ampulla comes together to form what? The lactiferous duct. Over here you have the nipple area. This is the areolar tissue. Here you have the fat. Okay, here you have the fat. At the back side you have the ribs. Okay, and the muscles between the ribs. That's all. That's all. So have that understanding. If at all you need, you just need to draw like this. So you have the alveoli, make multiples of them. Okay, alveoli are going to come together to form the tubule. Tubule, my dear children, will be coming together to form the duct. You're going to get the duct from here also. You are going to get the duct from here also. The ducts, my dear children, are going to come together to form the ampulla, and the ampulla, all the different type of ampulla which are there, it will come together to form what? The lactiferous duct. And this is whole happening in one lobe, second lobe. You can just put it down there. It's easy. It's easy. Remember this flow. Cool? Cool, ma'am. Now, schematically represent incomplete dominance by taking inheritance of flower color in Snapdragon as example. So, incomplete dominance means what? We have studied capital T and small t. When it comes together, capital T is dominant, it will be tall. But over here things are not the same. You have capital R, which is for red, and small r, which is for white. Okay, suppose I have a genotype like this, capital R, capital R. What color will the plant be? Ma'am, it will be red. Okay, so I'm not writing the red over here, I'll be writing it over here. It is red. Ma'am, then small bar will be white, right? Yes, it will be white, obviously. Now when you fuse them, what do you expect? Let's see. This and this fused, capital R smaller. This and this, capital R smaller. This and this, capital R smaller. This and this, capital R smaller. Ma'am, R is there, R should dominate. But you know what? It is not able to completely dominate over it, which is why none of them are going to be red. Rather, they are going to be pink in color. This is what? Incomplete dominance. The capital R, the dominant allele, is not completely dominant over the recessive allele. Okay, Mendel was like, not Mendel, people like what should we do? Let's try one more thing. Let's try to cross both of them. So let's try to cross capital R small r. Okay, capital R small r and capital R small r up over here. I make the box like this. Let's see what do I get. Capital R comes together. Capital R small r, capital R small r, and small r small r. One is red because both of them are capital R. One is going to be pink, another one is also going to be pink, and one is going to be white in color. So I get a ratio like this: capital R, capital R small r, and small r small r. How many? 1:2:1. What is the genotypic ratio? 1:2:1. What is phenotypic ratio? How do they appear? One is red, two of them are pink, and one is what? White. Phenotypic ratio also remains to be the same. It is 1:2:1. This is incomplete dominance. Snapdragon, or you call it Antirrhinum majus. So over here you see, you have to write it like this: phenotypic ratio. What does 1 stand for? Red. 2 stand for pink. 1 stand for white. Similarly, genotypic ratio, what does 1 stand for? Please mention like that. Please tell gametes are there. F1 generation is being formed. This is the parent and this is the gamete. Then F1 generation. Then you cross them. Okay, in Punnett square you can do that, and that's all. And please write the phenotypic and genotypic ratio. Cool? Cool, ma'am. Now, explain Griffith's transforming principle experiment. What is Griffith's transforming principle experiment all about? So, Griffith was in a search of finding the genetic material. So, he used one very famous bacteria that causes pneumonia, and that is Streptococcus pneumoniae. Now, Streptococcus pneumoniae has 2 strains. Okay, 2 strains. One is the R strain. R stands for rough. And the other one that we have is the S strain. S stands for smooth. Why is it smooth? Because the S strain, my dear children, has an extra covering made up of a polysaccharide. This gives it smooth appearance, and not just that, it makes it virulent. That means it is a troublemaker. It can kill organisms. If at all any organism have S strain, they can be killed. R strain is the non-virulent form of the strain. Now the experiment was done on the mice. What was done? So the mice is taken over here, or mouse is being taken over here. You put the live R strain, the one that does not cause any trouble. The mouse lived. Very obvious. Live S strain is taken and put into the mouse. Mouse died. Not at all predicted. Is it? No, it was also predicted. We know it's a virulent form, ma'am. Okay. Something that was not predicted is going to happen over here. Heat killed S strain. That means I treated this S strain with some heat and I killed it. Basically, I say heat killed S strain. I am putting in the mouse lived. That means something was getting damaged right over here, which is why probably it was not able to show its effect and the mouse was able to live. Okay, so I get to understand that this and this are making the mouse live. This makes the mouse dead. Fine, let's combine them. When both of them were combined, heat killed S strain and live R strain, the mouse still died. And that's where something unexpected was happening. He was like, how come that can happen? And then he also saw one more thing. In the mice, he found the live S strain. Did he put the live S strain anywhere? No, but he got it. He was able to retrieve it from the mice. So he was like totally confused. He said that something is getting transferred from here to here, converting the live R strain to what? S strain. And he called this thing as transforming principle. He called this unit as transforming principle. but he didn't know what was it. Was it a protein? Was it a DNA? Was it an RNA? Nothing. He got until here, then Avery, MacLeod and McCarthy came into the picture and then told that DNA is something that is getting transferred. But still they were not being accepted, which is why we have Hershey and Chase experiment. Finally it was proved that DNA is the genetic material. So this is Griffith experiment. You have to put the same effort for Avery, MacLeod, McCarthy and same efforts for Hershey and Chase experiment as well, even if in the model question paper if it is not there. I've told you, they give you hints. You have to understand the hints. Similar kind of questions could be coming up. So please have the idea of it. Now, this is the exact thing in a written format. Nothing else is there. Whatever I have said is put over here. So remember, it's a polysaccharide capsule, as in a carbohydrate capsule being made in the virulent form of estrine. Now, what are the functions of I gene, lac A gene, lac Z gene, inducer, lac Y gene? That is a question. That means you have been asked about what do you call lac operon. Do I have a picture of it? No. So when I do not have a picture of it, I have to make a picture of it. So it looks like this. Okay. You have a promoter region. You have an I gene. This I gene is what? Regulatory gene. Okay. So this is a promoter for this particular gene. What does it do? It regulates. What is it regulating? How is it regulating? By basically producing a protein that is known as repressor protein. Okay. Then you have a promoter region and you have an operator region for the next structural gene, which consists of Z gene, Y gene and A gene. This is the promoter and operator. And Z codes for beta galactosidase, which breaks down lactose to glucose and galactose. Y codes for permease, which increases the permeability of the cell. A codes for transacetylase, which deals with any toxic substances which is being formed. Now you might be thinking, what is ma'am saying? She's going on teaching herself or what? So listen to me carefully. I'll tell you one thing. This whole thing is there inside the E. coli. Inside the E. coli, it's a bacteria. And E. coli needs energy to survive. Where do you get it energy from? From the medium where it is being grown. Now medium can have glucose or it can have lactose or anything out there. If there is lactose, the lactose is taken up and beta-galactosidase breaks it down. Suppose lactose is not there. Do you think we still need to produce this thing? No. Do you think we need to increase the permeability to make it lactose inside? And if lactose is not there, how can it come inside? So basically, we do not want any of these genes to be transcribed. Okay, we do not want any of these genes to be transcribed or to be formed. From DNA to RNA, transcription should not be happening. So what do? The repressor protein right over here goes and binds on the operator region, such that the RNA polymerase, which will come to what do you call transcribe, will not be able to move forward and would not be able to do the transcription. This is known as switched off condition. Switched off condition. Now suppose, my dear children, over here we are providing lactose. We are providing lactose. Lactose will get inside. And what does lactose do? It acts like an inducer. Lactose acts like an inducer. Why? It binds with the repressor protein. So lactose, which is there, it binds with the repressor protein, and now the repressor protein is not available to bind on the operator region. It's free. RNA polymerase can do the transcription. So over here, that is why it is known as an inducer, because it helps in switching on the lac operon. Over here, it's not just lactose but also allolactose. It can be either lactose or allolactose. Now you might have a question, ma'am, how did lactose enter if at all we were not producing permease? My dear children, if at all whenever we say it's switched off condition, it's not completely off. There is still production of some amount of enzyme, which is why lactose is able to get in. Now once lactose has gotten in, obviously it will increase the permeability more because permease can be transcribed now. Similarly, lactose is later broken down to glucose and galactose, giving energy, and transacetylase deals with any toxic substance out there. So this is the story. Take a screenshot of this because you might not get this particular thing because it's an added slide out there. Okay. So over here we are there. I gene, regulatory gene, is for producing a repressor protein that binds to the operator and switches off the lac operon in the absence of lactose. What is the use of LAC-Z gene? Codes for beta-galactosidase, an enzyme that hydrolyzes lactose to glucose and galactose. What does LAC-Y gene do? Codes for permease, which increases permeability of the cell membrane to lactose, allowing lactose to enter the cell. Yes, LAC-A gene codes for transacetylase, which helps in detoxification of toxic byproducts which are being formed. Inducer is allolactose or lactose, binds to the repressor and inactivates it, thereby allowing transcription of the structural genes, switches the operon on. This is the funda. Okay, so this is the funda. You need to remember it. Done? Done, ma'am. Now, briefly explain the uses of transgenic animals. What is the use of transgenic animals? Any guesses out there? Let me drink some water. Transgenic animals means basically I am inserting a gene into an organism. I'm transforming the organism for my own purposes. This is transgenic animal, as in genetically modified animals. Just give me a second. Yes. So over here, the first, just a second. Yeah, the first one is basically to understand, understand the normal, it's normal physiology and development of an organism. That is one thing. For example, I have insulin-like growth factor, but I do not know what effect does it show inside the body of an organism. I insert it inside a mice and then I see the effect. I get to understand what are the things happening inside the body of the mice, and basically I get to understand that this could also happen in my body. These are the effects. So for understanding the normal physiology and development, we need transgenic animals. Second thing, for understanding the diseases. For example, I cause a mutation in the genes of a mice that causes cancer, and now I see what are the things that are happening inside the body of that organism. And probably I can also give it some medicine and see how does it respond to it, what is the work. So that comes a secondary thing, but basically for understanding how a disease works inside our body, what are the problems that it causes. When we know the problem, we can find the solution as well. So what are the diseases that is usually being asked? Please remember, it's cancer, cystic fibrosis, arthritis, and Alzheimer's. Okay. Cancer, cystic fibrosis, arthritis, as well as Alzheimer's. These are things that you should be keeping in your mind. Now, biological products. You can also get biological products from transgenic animals. This question is super repeated. First biological product that we get is alpha-1 antitrypsin. This is a protein, not an enzyme. Listen, enzymes are protein. Enzymes are made up of protein, but it can stay in the form of protein also. Proteins can stay in the form of protein also. They need not become enzymes. You get it? So it's a protein, human protein, that is alpha-1 antitrypsin. Why do you think we need it for? For treating emphysema. Emphysema is a respiratory disorder, and for treating it, we need alpha-1 antitrypsin. Okay? Used for human protein production, that is alpha-1 antitrypsin for emphysema. Now, there is one more thing. A transgenic cow, a cow was made to be transgenic. A gene was inserted which made the cow produce human milk, which is highly nutritious. What is the human milk which is made up of? Alpha-lactalbumin. Alpha-lactalbumin. So Rosie, transgenic cow, produced nutrient-rich human-like milk that is made up of what? Alpha-lactalbumin. Okay, that are the two products. So one is alpha-1 antitrypsin, which is used to treat emphysema. The second one is alpha-lactalbumin, and this is produced by a cow. The other one was produced by some other organism. The name of the organism is not mentioned in your NCERT. The next use that we have is vaccine safety testing. For example, I have made a virus. Now, obviously, before putting into an organism, a human being out there, put it into some other organism and see how does it affect. Is it a problematic thing? Is it causing any troubles out there? That is known as vaccine safety. So during polio vaccine formation and all, these things were being done. Now the next thing, chemical and toxicity testing. So what do we do is that we make the organism sensitive to even little amount of these chemical drugs. So when it is given to them, they show They show what are the effects. So we get to understand a person who might be sensitive to certain things, how do they react? Okay, what are the chances of what reaction could they give? You get it? Their body could give. So that can be understood with the help of transgenic animals. I totally understand. I always say in every class, this is not really great, and not organisms are not meant to be treated like that. But yes, this was the case previously. Now it has been reduced to some extent out there. Initially, I remember my brother in his school and all, there was cutting of frog and these things and that. When I came into 12th grade, there was nothing like that being done. So I'm expecting some things have already evolved in a better manner. So probably there might be some other substitutions for this. But anyway, this still persists. That is also true. You need to know that. So these are the uses of transgenic animals. Now we have, what is mutualism? Explain any 4 examples of mutualism. So what is mutualism? Mutualism is when both the organisms which are interacting, they get the benefit. Both benefit each other. For example, lichen is a mutualism shown by algae and a fungus. Algae and a fungus. So algae basically provides food by photosynthesis. They have the ability to do that. Fungus provides protection and absorb water as well as minerals. Fungus, as I said, they have root-like thing, rhizoids. They get into the areas and provide the nutrients and everything out there. Apart from that, they also provide protection over here. So that is about lichen. Both of them are helping each other. Mycorrhiza, we already discussed about this, fungus and the plant roots. Fungus helps the plant in absorption of water and minerals, especially phosphorus, and plant provides the food to the fungus. Now, rhizobium bacteria and leguminous plant. Rhizobium bacteria is a nitrogen-fixing bacteria. Leguminous plants, as in pea plants. Both of them, my dear children. Similarly, rhizobium fixes atmospheric nitrogen, and over here plant provides shelter and nutrients. Whenever plant is involved, they're obviously going to give out organic matter out there, okay, or nutrients out there. Fig tree and wasp. Fig tree and wasp. Wasp basically helps in the pollination of the fig flowers, and the fig, my dear children, provides the safe space for the wasp to lay their eggs. So that is how they help each other. Also provide the food. Also provides the food to have. Pollen grains and everything, they are not totally being transferred. Some of it is being eaten also. Pollen is highly nutritious, right? So these are the things that you need to remember as examples of mutualism. Okay. Now we have 3 more questions is what I believe, and these are one question you have to attempt in this category, in this 5-marker question category. Identify the genetic disorders given below. Sex-linked recessive disorder occurring in about 8% of males and only about 0.4% of females, leading to a defect in the eye, in the cone of the eye. So cone of the eye means it has to do something with the color blindness. It has something to do with the color blindness, my dear children. Okay, so over here, what is exactly happening? Listen to me. Why are males being affected? 8% of males and only 0.4% of females. Why? Because this is a sex-linked recessive disorder. That means, suppose the female have one of her X chromosomes being mutated. She will be just a carrier. She will be just a carrier. She does not have the disease. Whereas in males, if one of his X chromosome is mutated, he will be having the disease. Why? Because there is no other X chromosome to overshadow the effect. You get it? So that is the reason. For a female to have the disease, you should be having both the X's being mutated out there. Now, sex-linked, so this is color blindness, okay? Sex-linked recessive disorder affecting a single protein involved in the clotting of blood. Another sex-linked recessive disorder, here what is happening, clotting of blood is being, disoriented. Because the protein is being missing. This protein is being not there. Why? Because the gene producing it is somehow mutated. What is this disease? It's hemophilia. It's hemophilia, where again the males are usually being affected more than the females, and my dear children, it's a life-threatening disease, not like color blindness. The males would not be able to survive until the time they can reproduce, which is why if they are not involved in reproduction, listen, from the male side you should get an X for a daughter to be born. From the male side you should get a X for a daughter to be born. Suppose mother is a carrier, father is having the disease, this and this should come together for the child to have the disease, right? But the male is not alive until that particular point of time to reproduce. Get it? Which is why females are least affected in this. Okay, so where are we? Yeah, so here a gene which is responsible for the formation of protein, that is responsible for clotting of blood, is affected. And this disease is hemophilia, where you will be having complete flow of blood if at all you get a small wound. Now, inborn error of metabolism, autosomal recessive trait, accumulation of phenylalanine. So this is inborn error of metabolism. That means it is inherited. It is autosomal recessive disease and accumulation of phenylalanine is happening. What is this disease? It is phenylketonuria. Phenylketonuria. Why is it happening? Because one enzyme, one protein, one gene that codes for an important enzyme, that is phenylalanine hydroxylase, is being mutated. Now, when phenylalanine hydroxylase is not being produced, the phenylalanine which is there in our body, it won't be able to convert into tyrosine. Usually PAH enzyme does it, phenylalanine hydroxylase does it, but it is now not there, which is why this is going to accumulate in the form of phenylpyruvic acid. Phenylpyruvic acid, and this accumulation causes mental retardation and things like that. Okay, please remember. So over here it is phenylketonuria. Apart from that, you also have other symptoms as well, such as this tyrosine, my dear children, is a super important thing for formation of melanin that is responsible for our pigmentation. So since melanin is not being produced, we have hypopigmentation, less skin color, as in reduced pigmentation, gray hair and whitish skin will be there. Now, autosome linked recessive blood disease, substitution of amino acid in beta globin chain of hemoglobin. What is this? This is sickle cell anemia. Why sickle cell anemia? Because substitution of amino acid is there. If at all you have studied sickle cell anemia, you know you had a gene that is GAG and it is replaced by U here. GAG codes for glutamic acid, but U codes for valine over here. So initially it was this amino acid, but now it is this amino acid, which is why the structure of beta globin chain is hampered, which is why the cell structure RBC, which carries the hemoglobin, that its shape is also hampered. It is now sickle in shape. That is why sickle cell anemia. Always remember A comes before U, so it was A first, then U. Always remember G comes before V, so it was glutamic acid first, and then it was replaced by valine. Okay, it is happening at the 6th codon position in the beta globin chain, not the alpha. Hemoglobin is made up of two chains, alpha and beta. The beta one is affected. The quality of is being affected, not the quantity. Quality is being affected. Now, autosome linked recessive blood disorder, reduced rate of synthesis of globin chains of hemoglobin. Reduced rate means you're talking about quantity. If some gene is mutated, which is why enough amount of beta globin chain is not being produced or alpha globin chain is not being produced, my dear children, you are affecting the quantity, and over here the disease is thalassemia. Thalassemia is a quantitative blood disorder. Sickle cell anemia is a qualitative blood disorder, and hemophilia is also blood disorder, but it is sex linked. Here both of them are autosome linked. So your answer for this question is this. Cool. Next. The flowchart shows replication of retrovirus in the host. Answer the questions given below. Fill in the missing data in the boxes labeled as 1 and 2. Labeled as 1 and 2, okay? Give an example for retrovirus. Name the disease caused by this retrovirus. Name the diagnostic test for the disease. Okay, fine, cool. Let's get ahead into it. So we have the retrovirus. Okay, retrovirus infects the normal cell. Viral RNA is introduced into the host cell. What happens after it is introduced? So usually we get DNA. DNA transcribes into RNA. That's how it works. But now the retrovirus is coming with an RNA and it wants to get inside our DNA. So it has to form a DNA. How can that happen? We have to do reverse transcription. Now the viral DNA is being formed over here with the help of reverse transcription. Who does it? Reverse transcriptase enzyme does it. Now we have got the DNA, viral genetic material in the nucleus of the host cell. Viral DNA incorporates into the host genome, gets into our genome. Now what? It will, obviously, when it integrates into our genome, it will make sure that it uses our machinery to form its viral RNA, to form its viral protein, to form everything which is important to it, right? And then it will all come together and the viruses will come out of the host cells and will infect the other things. So over here, your answer for this is viral DNA is formed over here. Okay, after reverse transcription, the viral RNA is converted into DNA, that is box 1. Box 2, viral components such as protein and RNA are synthesized. That is this. After they incorporate into the host genome, obviously they use our machinery to form the viral proteins as well as the RNA. The reverse transcriptase enzyme is also a protein. They bring, okay, So retroviruses are super in that. So give an example of a retrovirus. It is HIV, human immunodeficiency virus. Name the disease caused by this, and that is AIDS, acquired immunodeficiency syndrome. Name the diagnostic test. You have ELISA, enzyme linked immunosorbent assay. You only have 2 tests for 2 different diseases. One is typhoid, that disease is the test is Vidal. The other one is HIV, the test is ELISA. Cool? Cool, ma'am. Let's go. Now this I assume is the last question. After that you just have one MCQ for the visually challenged children. So give reasons for the following. Restriction enzymes are called molecular scissors because they cut the DNA out there. They are the one that are responsible for cutting the DNA at specific sequences. Give an example of EcoR1. EcoR1, my dear children, has a restriction site of 5-GAATTC3-. It makes a cut between G and A. Okay, now the next is DNA ligase is used in genetic engineering. Why? Because when we cut things and we put things, suppose we are putting it inside a plasmid, we need to make sure that the genes are joined. For that we need DNA ligase because it joins the 2 DNA fragments by forming phosphodiester bond. Phosphodiester bond is there between the nucleotides, right? So that has to be formed with the help of this enzyme. Cool? Now, chilled ethanol is used in DNA isolation. Chilled ethanol, why? Because when you put chilled ethanol, the DNA precipitates around, my dear children. That's the thing, or floats around. That's the thing. So because DNA is insoluble in cold ethanol, it is insoluble in cold ethanol, it precipitates out from the cell extract, making it visible and easy to collect. And the process of collection is known as spooling. Now, smaller DNA fragments move farther in a gel slab. Why? Because, my dear children, if at all you have seen gel electrophoresis, they move. The DNA does not move on the surface, they show sieving effect. They move within. They move within. And when they move within, my dear children, the smaller fragments which are there, they find less resistance to move. That is why they move farther. So, because during gel electrophoresis, smaller fragments face less resistance from the gel matrix and therefore migrate faster and farther towards the positive side of electrode. Remember, DNA is negatively charged, they move to the positive side. Positive is known as anode. Anion is negative, but anode is positive. Okay. Now, thermostable DNA polymerase is used in PCR, polymerase chain reaction. Why do you think in polymerase chain reaction you use thermostable polymerase? Because PCR is happening at high temperature. Denaturation happens at 95 degrees Celsius. Annealing happens at almost 50 to 65 degrees Celsius, and the last one, which is extension, that happens in 75 degrees Celsius. Obviously, there should be an enzyme that could be stable at that particular temperature, right? That's why we take the enzyme from a highly thermostable organism, that is Thermus aquaticus. It's a bacteria. It's highly thermostable. You take from there. Okay, so Thermus aquaticus is the thing. So, because enzymes like Taq polymerase can withstand the high temperature used to denature DNA strands, allowing the PCR cycle to continue without the enzyme breakdown. Here I have some pictures. So when you put chilled ethanol, as you can see, the DNA moves out. In gel electrophoresis, the smaller ones move farther, the larger ones move slower. If at all the question is who moves first, we do not know who moves first. But anyway, at the end of the race, the smaller ones are over here. So do not go and write the smaller ones move first. We do not know that. Who are the one who it can be larger one, it can be smaller one. But the one that moves farther is the smaller one. DNA ligase, as you can see over here, is like a glue. Listen, this cute DNA ligase are the glue, all the things that is being written over here. So ligase is the glue word. Cool. Now, the restriction site for EcoR1 enzyme is, what is the restriction site, my dear children? 5-GAATTC3-. Don't go and write this answer, it is different polarity. Here, polarity is not even mentioned. Your answer is does this, and it cuts between G and A. So, over here, this is your answer for the question. And with this, we end today's model question paper. I hope, my dear children, this video was helpful for you. If it was, share it with your friends, and as I said, subscribe to the channel right now. Okay, and wishing you all the best. If you have any doubts, put it down there. I am here to help you out, and you guys are the best. Okay, so bye bye, take care.